Hello, good evening to everyone participating from Sri Lanka, and a very good morning to Professor Gomika Udugamasooriya, our invited speaker from the University of Houston College of Pharmacy, USA, and the other participants, if any, from the USA. On behalf of the Department of Chemistry, University of Peradeniya, and as the head of the department, I'm pleased to welcome all of you in this beautiful evening to the 19th Inspire webinar series conducted by our department. So we have a very special speaker, Professor Gomika, today to just give you the 19th Inspire speech. So then Gomika was a product of University of Colombo, and I'm happy to say that he has also obtained his PhD from the same university from where I obtained my PhD, that is Wayne State University in the USA, and I'm so proud of that. And currently, he is a renowned researcher in drug discoveries. So Dr. Subhashini Gunatilaka will give you a nice introduction about him. Gomika, if I brief you the intention of having this Inspire webinar series, we really wanted to make our students aware what he or she could be in the future and how she or he could contribute to science in future. Therefore, giving them a chance to listen to scientists like you would help them identify the hidden scientist in them. So that's the main target. Gomika, thank you so much for accepting our invitation despite of your busy schedule, and also agreeing to make this presentation early in the morning in the USA. So I'm sure this lecture will be a very fruitful one for everyone. So without taking much time, I would like to invite our Dr. Subhashini Gunatilaka to introduce Professor Gomika Udugamasooriya to the audience. Have a pleasant evening, everyone. So it's over to you, Dr. Subhashini Gunatilaka.
Thank you, Madam. Good evening, everyone. It's my great pleasure to introduce the speaker today, Professor Gomika Udugamasooriya. Professor Udugamasooriya obtained his BSc Chemistry Special Degree in 1999 from University of Colombo and PhD in 2006 from Wayne State University, Michigan, USA. Then he completed the postdoctoral fellowship at University of Texas, Southeastern Medical Center, Dallas, Texas, and then joined the faculty at the same university in 2009. Then in 2014, he moved to University of Houston and MD Anderson Cancer Center, Houston, USA, which is one of the world's largest medical centers, and currently serves as a tenured associate professor of medicinal chemistry. Professor Udugamasooriya is directing a multi-millionaire cancer research program, mainly focused on cancer drug lead discovery and diagnostic tool developments funded by research grants from National Institute of Health, Department of Defense, Cancer Prevention and Research Institute of Texas, Innovative Research Award from Rheumatology Research Foundation, and various other sources. He has published 36 research articles so far in world's topmost journals, including Nature Chemical Biology, Journal of American Chemical Society, Clinical Cancer Research, and European Journal of Medicinal Chemistry, etc. He also presented over 75 abstracts and gave over 45 invited seminars in US and worldwide. He introduced a novel two-color high-throughput drug discovery technology and has 8 patents so far. He is an editorial board member and a reviewer of many international scientific journals. He is a member of grant review panel in US Government National Institute of Health, private foundations like Kirk Foundation, and multiple international institutes. He received numerous awards, including Chancellor's 15-5 Scholar, Faculty Excellence in Research from University of Houston, NIH Cancer Excellence Career Development Award, and in his undergraduate career, graduating with gold medal in chemistry. Professor Udugamasooriya is the chair of the Graduate Education Committee at University of Houston, and he has been an advocate for many younger scientists and engaged in numerous cancer building career building seminars and workshops both in US and Sri Lanka. In addition, Professor Udugamasooriya is an artist and a philosopher as well. He is actively engaged in conducting seminars and workshops worldwide on cancer prevention and research, highlighting the value of practicing holistic medical approaches that includes developing good eating habits, building physiological strengths, positive thinking, in addition to conventional cancer treatments. Today, he is with us in our 19th Inspire webinar to share his experience in developing technologies to expedite the drug lead discovery. Without further ado, let me invite Professor Udugamasooriya to start his talk. Over to you, sir.
Good evening to you all. I hope you can hear me clearly, right? Thank you so much, Subhashini, for this kind introduction and also the invitation, and of course, Professor Gani Henege for also this invitation. I have been told that this is Inspire webinar series, so I have been asked to inspire you, and I'll try my best. And give me 1 second to share my slides. I hope you can see my slides clearly, right? Okay.
Yes, Professor.
Okay. All right. Okay. So when I was asked to inspire you all, and I thought rather than talking about these common words like, okay, you have to be energetic, enthusiastic, or whatever, I decided to tell my story, which has a lot of take-home points and many things to learn through my career. We are chemists. And if you all here to see chemical structures and reactions, I'm going to disappoint you. I am a chemist, but what I'm trying to tell you today is, as chemists, what are the things we can do to change the world? And what are the things, how we can utilize our power to apply this, the beautiful chemistry in many different areas. And of course, my area is in the biomedical field, so I will talk about on that area. So that's why I kept the title as Wisdom Beyond Chemistry. Okay, and then later of my talk, I have a lot of data slides, but I will go through them very rapidly, so please bear with me. I'm trying to give mostly the messages throughout my talk. Okay. So as chemists, we are very good at dealing with molecules, whether small or big. We synthesize them, characterize them. But how many of you all have thought about what are the impact we can bring in to the society and also to change the world, the application? So for example, I'm talking about, like I said, the biomedical applications of the chemistry, these molecules. I'm going to show you a lot of projects on this area. And also, when we get into our lives, not only in academic life, we should always have goals. And when we set up those goals, we should have a straight path. There are so many different ways. There are a lot of distractions, stop signs, roadblocks, but we need to move forward on a straight path to reach our goals. And then as scientists, I always encourage you all to think always this way. You need to target big, but it's very, very important to think simple. And I will show you a lot of examples how we can do that. And also stay focused. This is very important, and not to look at like these very complex ways, but we can find answers for many big things in a very simple thinking. Okay. So it all begins here, my story. Actually, the high-level academics in the University of Colombo. But before that, I was also doing a lot of interesting things, projects in school. I went to the SNNI College. And even as a small kid, I can remember I was really interested about research, science, and then at that time, the kind of the picture come to our mind, a lab means something happened underground secretly. So I can remember I was like having dig some areas in the backyard, then go there and do things. So anyway, so when I came to University of Columbia science faculty, I missed medicine, few points, that's fine. I didn't give up. So you all know, after the second year, we have a chance to do the special, and I got selected for all three in the biology area. Of course, I'm a chemist, so I choose to go for the chemistry special. So in our first lecture, which was done by Professor Dilip De Silva, and I think I don't need to introduce Professor Dilip De Silva. You all know, one of the best organic chemists produced by Sri Lanka. And with his reputation, and also we heard like he's asking students to come to the board when he teach. So everyone was sitting behind, I mean the back row. So I wouldn't say I'm fearless, but I was sitting in the front. So he started the lecture and he started getting into this esterification reaction under acidic conditions, and he started asking questions. So no one was answering. So I started giving some answers. So immediately asked me to come to the board and asked me to think about writing the mechanism. So I wrote this lone pass and I put the arrow correctly. And as you see, he started smiling. And from that exact moment to the moment I'm talking here, we had a very good, strong relationship built. And I was enormously benefited through that. And he helped me not only in the academics, but even in my personal life, doing TOEFL, getting to US, there are a lot of things happen. Why? It's because I came forward. And if I was sitting in the back row, I would have still got the 4.0 GPA, but I wouldn't have sitting here and you wouldn't have invited me to give this talk. So the take home point is don't be shy. I mean, one of the weakest wrong things that we do is we don't come forward. So you need to come forward and then it will open up a lot of opportunities for you. Okay. And then I moved to Wayne State University. I'm very happy that it's the same place that your department chair, Professor Gani Henage, also graduated. So there, I met my thesis advisor. This is our first meeting, and you can see how I was very energetic, enthusiastic, and this was my first question. So from the very beginning, I had a very interest. I mean, I wanted to develop drugs and medicines, right? So I was asking, can we develop a drug? And he looked at me like an alien fallen from the outer world. And anyway, he kindly explained to me, okay, we cannot develop drugs in the academic places, so it has to go through a lot of process, of course, and I learned that. So in the modern drug discovery process, uh, it has to go through this very tedious process. So from about 10,000 compounds we identify initially, they are called drug leads. So, and then only about 250 getting into the animal level, and then only about 5 getting into clinical trials, and then become one drug. So out of 10,000 compounds, only one become a drug. And then we have to spend about $1 to $2 billion, and we spent about 10 to 15 years to do this. And on this shaded area, we all almost always need help from the academic, the industry. So this is a huge process. And I'm going to tell you my story, and I have stories within that. And also I want to mention something happened to me over past few years. Maybe some of you might have seen, there was some Facebook uh, post saying I found a cancer drug, and that is wrong. So please don't follow those. And drug developing a real drug is a huge process. Of course, I have multiple patented compounds going through this process and even getting into this area, and I'll talk about them. But I have not developed a drug. I want to be very clear on that. And I never said that, and I don't know how those posts came. Anyway, all right. So getting back to the my story, this is my PhD work, and this is one of drug lead that I developed, and we solved the crystal structure. And I'm not going to focus on this. And after my PhD, I did my postdoctoral fellowship in University of Texas Southwestern Medical Center. This is one of the topmost medical centers in the US, and it harbors the largest number of Nobel Prize winners actively working. There are about 2,000 faculty, permanent faculty. So there, again, this is my first meeting with my postdoctoral advisor. And you can see the same story. But by this time, I was more practical. I know what are the things we can do, but still I was focused. I didn't give up. And I was asking him, can we accelerate, at least accelerate the drug discovery process? So he asked me, what do you want, whom do you want to become in the future? And I immediately said, I want to become like you. And then he told me, very good. He showed his research program. He took about 2 hours and show me his whole research program, and then told me, okay, go back and develop something new, a new project, and then that can be done in my lab, and then come back in about 2 to 3 weeks time. You all know we are very excited, and I went back and I put together some projects and came back to him within 2 to 3 days time. And then the first question he asked me was, I asked you to come back in about 2 to 3 weeks, and why you came this early? And then I thought, okay, this is the moment that I can get some points and show my colors. And I showed him, okay, I have some projects. And he looked at, and he again asked me, what do you want to do in the future? And I said, I want to become a person like you. And then he just simply threw my projects into the dustbin. And he told me, if you want to become a person like me, an independent investigator, you need to bring brand new projects, new ideas. And then I realized why he has asked me to take 2 to 3 weeks time. All right, I went back, and still I want to work on this area. And I started looking deep into what is happening. Now, here actually I'm showing the research, the R&D expenditure over the time compared to the drug output. So of course it is the data is missing a little bit here, but the research expenditure has sharply increased, but the drug output has not really improved. This is a problem. This is a question for me. And then here I'm showing the same thing. This is actually showing new drugs developed per $1 billion. You see, in early time, there were many drugs came in, but over the time, it's basically now only one drug come out per $1 billion. This is a problem. So I was staying focused, and I want to work in this area. And I started looking at what is this conventional drug discovery process? Basically, actually the drug lead discovery process. So what happened is, biologists first identify the disease biomarkers. These are the areas that we want, these are the biomolecules we need to target. Enzymes, receptors, signaling molecules, whatever. And then chemists come and develop drugs to target this. Then only you look for off-target effects, whether it will attack normal cells, because the biological system is very complex. So this is where lots of compounds get lost. From about 10,000 compounds, only one become a drug, because 99.9% of these drugs get off-target effect. It is true, right? This is a huge human body. It's a hugely complex system. So when I was thinking about this, I thought, to answer this question, if you want to cut down the time and resources spent here, why don't we bring these two together in the initial drug discovery process? Bring this normal cell or the off-target effect factor into the initial drug discovery. So that is my concept. That's what I thought. And then again, like I said, we are going to do a huge, big thing, but it's a very simple thing. We bring this factor also here and then stay focused. All right. So we are actually it will not solve the way we are doing, not solve all the problems, but at least to a certain level. So we are targeting cell surface biomolecules, biomarkers, and there are two major tools. One is the compound class we are using, that is peptoids, and then this two-color technology that I developed. Okay, so in drug discovery, there are two major ways we can do. One is the structure-based drug discovery, rational design. So it's like you cut in the key to the keyhole, you work with one molecule, and then you target it into the biomarker we want to. For example, if you want to inhibit enzyme hormone, you cut the key and then work. This is work on one molecule at a time. And the other method is combinatorial high throughput screening, where you bring all the keys and ask the keyhole to pick the best one, fitting one. That's the idea. So large number of libraries. And then actually we can use those compounds to attach imaging agents and use for diagnosis. And there are a lot of applications. So this is the area we are working on. And this is what the modern drug discovery is mostly based on, high throughput screening. Okay, so here actually I'm showing three major clinically approved drug classes: small organic molecules, big antibodies, and then peptides. So the molecular class we work are peptoids, which is originated from peptides. And what is the difference between peptide and a peptoid? I think you all know peptide is a chain built with the amino acids, and peptoid, this R group, which is in alpha carbon of this normal amino acid, is moved to amide nitrogen here. Because of that, these molecules become much more serum stable. Peptides are not good because if you take it orally, it will break down by the enzymes. It doesn't work as a drug. And these peptoids are non-immunogenic, serum stable, highly cell permeable, highly diverse, and there are synthesis is very rapid and economical, and there are a lot of good characteristics as peptoids. Now, this is Professor Ron Zuckerman. He is the father of the peptoids. He invented this molecular class in the early 90s. And then my postdoc advisor, Professor Thomas Kodadak, showed these great qualities in the early 2000s as a biologically applicable molecule. Now, this has been emerging as a very interesting molecular class in drug discovery. So I'm very, very lucky to work with both these fathers of this molecular peptoid compounds. Okay, the synthesis. We do this synthesis on solid phase resins. So in peptide synthesis, we add amino acid at a time and then cleave it off. But in peptoid synthesis, in order to add one residue, we go through these two simple steps. There are reactive centers ready to react on these resins. And then this bromoacidic acid brings the two carbon, these amines substituted, and then it will add one residue. We go through the same reaction in the next residue, the next step. So each of these steps can be done microwave assisted synthesis, less than 1 minute. And then this R groups comes from an amine. That means we can go to thousands of different amines and bring the huge diversity compared to peptides. Okay, I mentioned we are working on high throughput screening, large libraries. So how do we develop that? Now let's take three amines called A, B, C. And we have resins, and then we couple A here as the first residue, and B here as the first residue, and C here as the first residue. And then we mix them, and then randomly separated them back into three reaction vessels. We get all these mixtures in these three vessels, and then we couple A here, B here, C here as the second residue. Now you can see already 9 combinations are there. How about having more amines, more steps? And that's what we are doing. This is actually 8 different amines, 6 steps. We get quarter millions of compounds. And these libraries can be developed within a few days' time in my lab. And we do that in microwave assisted synthesis. This is another library with about 400,000 compounds. Okay. Once we have these libraries, it actually build one bead, one compound. Now, each bead carry a unique sequence with a lot of copies. Different beads have different compounds and millions of compounds. So how do we find the best compound for our targeted protein? So the way we do that is, let's say we want to inhibit a certain enzyme of protein. And then we engineer that with a tag and expose to the library. And then when it binds to a compound on one of those beads, that bead get light up. And so we pick it up and identify the compound. And that compound become a drug lead for that protein. So this was the standard drug discovery process, on-bead drug discovery process there for about 2 decades. Now remember, I mentioned the problems, the concerns. So the specificity is not guaranteed. This compound can attack anywhere. We don't know that. So, and also targeting cell surface receptors is not that easy because you cannot engineer these proteins easily for cell surface receptors. So to resolve this problem, I came up with this idea. This is where I developed this assay, where I let that cell surface receptor to be sitting in the natural condition in the cell and expose the whole cell to the bead. But not only that, with this very important control, which is the parent cells. So basically, we take divide the cells into 2 groups. One has the receptor that we want to target and the other don't. So ideally, the only difference between these two is this has the receptor, this don't. And then actually we use this VEGF receptors as this model system. So VEGF receptors are receptors found in blood vessels and very important in cancer progress, where when the tumors are growing, it cannot grow beyond few millimeters without the blood supply. So what happens is it secretes this VEGF hormone, it binds to these receptors and triggers signaling to form blood vessels towards the tumor. So if you want to block this, we want to target this receptor. There are drugs. So we use this system. So we had 2 cell groups. One has the receptor, the other don't. And then I did very simple thing. This is where I'm telling simple, simple, simple. So kindergarten thing. I stain these red cells in, the receptor positive cells in red, parent cells in green, mix 1-to-1 and expose to the library and looking for beads that bound only red cells, not green cells. That means the compound on that bead only targeting our receptor that we want to target, in this case VEGF receptor 2, and it doesn't bind to any other cell surface molecule off target. And if it binds, they are found on green cells, and we see both red and green bound. And you can see both red and these are the actual images. So both red and green bound. So we eliminate them, and we only look for red cell bound bead. So these are the actual fluorescence microscopic images of the screen. We did 400,000 compound screen, and we found only 5 of these very specific hits. And we found about hundreds of these. And if we didn't have this green control, we would have found, isolate them as drug leads, and then spend many years. And then later we find they are non-specific. So what this technology does is it actually eliminates the non-specific off targets at the very first screen, very first day of the screen. So we find we can do now in my lab, we do 100,000 compound screens per day. So we eliminate all the non-specifics and find only the highest specific drug lead. Okay, and then we get this bead, identify the compound. So this is again the take-home point. We did a very big thing. We resolved a problem that has been there for 2 decades, but very simple kindergarten 2-color approach, right? So that's why I say think simple. If you want to touch your nose, do that directly like this. Don't try to do this way. And I was not looking for multi-million instruments, the big things. I just looked this very simple way and it resolved the problem. So as researchers, as scientists, this is what you need to think about very carefully. Okay. So this is one of the compounds that we identified. I'm showing a very all the summary of the therapeutic applications, a lot of papers, and about this compound blocking the tumor growth, the blood vessel formation, and the angiogenesis. And then we use them for diagnostic purposes as well. So this is kind of a snapshot of what we do after we finding these compounds, applying all the way to the therapeutic and diagnostic area. Okay, so after my postdoc, I was offered, actually I was in the getting into the independent career, and I got multiple offers, but UT Southwestern also offered me a position, so I stay focused. And since I developed this, my postdoc advice also very gladly let me take this technology forward, and I started my first lab in the UT Southwestern, and then I moved to Houston, where to the University of Houston and also MD Anderson Cancer Center, and all together with some other institutes, it formed the Texas Medical Center, which is considered the largest medical center in world. So these are some of the pictures of my lab, and I have my own mass specs and the automated synthesizers, the spectrophotometers, fluorescence microscopes, and chemistry side, the the cell culture side, and there are a lot of instruments. So let me talk about several projects as examples. And the first one I'm going to talk to you about a very recent project. We work on the pandemic and targeting COVID-19. So I think you all know by now how the virus infect SARS-CoV-2 through the nose and different areas. And the mechanism is it actually binds to a receptor called ACE2 receptor, angiotensin converting enzyme 2, ACE2. And then the way it binds to this ACE2 receptor on our epithelial cells in the nasal cavity through this S protein, spike protein of the virus. So the simplest thing we can do is put a roadblock there, right? So develop a drug that you can block it. So and then it will actually become a molecular version of the face mask. So we can develop into nasal spray, so eye drops. So that would have been a simple idea, but it has not happened. Why? So if we try to target the virus side, the S protein always mutated, right? So with these mutations, you cannot keep the drug binding. So then you need to target to ACE2 receptor. So this is a SARS virus. All the SARS viruses use this ACE2 receptor as the main target. So that means 2 decades we know the mechanism, but there's no drug developed even targeting ACE2. Why? So this is what very first thing I thought. Okay, so this is the complex structure, the crystal structure of the ACE2 binding to this virus S protein. So this is the interface that we are going to block, put in the face mask. But I want to bring your attention to this area of the receptor, the enzyme. What is this ACE2? It is not there to virus to come and get in, but it is actually a very important receptor that actually control our blood pressure. It actually lowers the blood pressure. So this is the active site of that enzyme. And this is where the virus binds. So ideally, we want to block here. So we want to target here. So there has been a lot of effort in developing compounds, but what happened is they all affect this active site, and then it becomes toxic for us. So we cannot develop a proper drug. That's why it was not there. But here's the thing. I told you, think simply. Okay, when I thought about this, although we cannot develop compounds to bind to ACE2 and then it become toxic, how come virus does that, right? Virus doing that very simply. So that means there is some logic behind. So Again, very simple thinking. What I thought was, how about we mimic exactly what has happened in the evolution? That means the very first day when the virus reached to this receptor, how would have it bound? So if you try to open a door using a very simple way, if you follow that, you can block that exact way. That's what I did. And I have my technology which can simply mimic that. Remember we have beads displaying compounds, just like the virus having this protein. So the story is we went for this two-color assay. We had the ACE2 receptor positive cells, the negative cells, stain in red and green, and mix. And then we are looking for red cell bound beads. That means the compound binding to ACE2 exactly as the virus bound. So that's what we did. We found two compounds, and then immediately we dimerize it. And also we showed that immediately it actually can bind and pull down the ACE2 receptor. And also we quantified the binding. Actually, for when we come to the data, I will go very rapidly because I want to give you the overall picture. So it bind actually a very good range, mid to low nanomolar. And then actually we show that our compound can actually blocks this virus human ACE2 interaction. And also, most importantly, we show that it actually blocks the virus enter into the cell. This is good. This has been shown also before, but most of those compounds become toxic. That was the biggest problem. So we spent a lot of time on validating that, whether it affects this active site of the enzyme. And here actually I'm showing there are no toxicity over the dose we given. And also here I'm showing the protein has not changed. The receptor is same, no effect. And here actually I'm showing in many different cells, the enzyme works as normally. No enzyme, the ACE2 activity has not been affected with our compound. All right, so let's go back and look at this interface. So it looks, we have found a compound. Now we did some molecular docking and to see where actually our compound lands. And we found very gladly, it looks our molecule shown in pink, it actually lands the same exact place overlapping this virus S protein binding. Now, this is actually putting a face mask into this, right? And also how I found, just follow the simple way how virus doing that. So these compounds are actually right now in hamster studies, and I'm not allowed to talk about the data because that's the company related thing. So, and then as a summary of this project, we wanted to block the virus binding to the humans, putting a blockage in the right at the entry doorstep. We named this as a molecular mask. It's actually putting a mask on that. And then these can be developed into nasal sprays or eye drops. So how did we do that? It's a very big thing, right? But very simple. Just follow what virus did, and we found these compounds exactly binding to that area without affecting this place. All right. So my main area is in the oncology, cancer targeted drug lead discovery. So we are working on multiple different areas. So single targeted drugs are not working because these biomarkers are very heterogeneous in humans. And then the signaling pathways we are trying to block are very complex. And also cancer stem cells, they are the king of the tumor, very difficult to target. And also there are no perfect ways to real-time therapy monitoring. So these are the different areas that main projects going in my lab. So I mentioned to you at the beginning, the conventional drug discovery is the biologists find the targets and the chemists come and develop drugs to target that, then only look for the normal cells. So I said, I don't want to do that. I want to shorten the drug discovery process and minimize the expenditure. So I brought this normal also into the consideration with the disease cell, and then we can target only the disease cell, but not the normal cells from the very beginning, and identify only the very specific compounds that not only find this way, actually doesn't, I mean, mostly the conventional drug discovery targets the protein biomarkers, but there are a lot of lipids, carbohydrates, so we can target them as well using our technology. Again and again, the big thing, but very simple way of doing this. Okay, let me jump into the second project. So this is actually unbiased targeting of cancer stem cells over the remaining cancer cells. Why? So these stem cells, there's a lot of debate about this, but our normal cells just divide, but the stem cells can not only divide, it can differentiate as well. So what happened in tumors, these stem cells are very strong. They can live, they can actually resist drugs. So what happened is usually the conventional therapy, chemotherapy, or even targeted therapy can get rid of almost all the other tumor cells, but these cancer stem cells remain and bring back the tumor. So if we can target these cancer stem cells, we can go for a complete removal of the tumor. But in drug discovery, we need to have a target, right? So we always need a target. These cancer stem cell biology has not been very well understood. They are very cunning, and even they have mechanisms to throw out the drugs. We don't know biomarkers. So the conventional drug discovery is stuck here. So there are not good biomarkers reliable. There are some, but okay. So this is an ideal place to apply my two-color assay. We don't need biomarker. We don't need to know. This is an unbiased approach, which is we take the tumor cells and separate them into cancer stem cells and also remaining cancer cells from the same tumor. And then stain these stem cells in red. This is in green, mix 1-to-1, expose to the library. We are looking for red cell bound beads. What it will say? First of all, the compound binds to something specific for cancer stem cells, which is not found in other cancer cells. So, and then this specific thing is a biomarker, specific biomarker found only in the cancer stem cells, which we don't know even. So we can pull down and identify those, and also we have a drug lead to target them. So this unbiased application of this technology do two things: of finding new biomarker and also a drug lead to target that at the same approach. So you can remember we started as a very simple idea, but we can do really big things here. So we found one compound. Again, I will go very rapidly through the data, and we show that it only binds to the cancer stem cells, and we show it pulls down the cancer stem cells. And then now we don't know the target. Like I said, this is not the conventional drug discovery. We are reversing it. So how do we find that? So we let our compound to bind to that cell surface target, and we trigger cross-linking using UV activated this benzophenone. I'm not going to get into the chemistry, you all know. So, and then we pulled that receptor out of the cell and identify what it is. So we did that, and then we found a protein called plectin as the target of our compound, which is specifically found in cancer stem cells, the most troubling cells. So we did a lot of validations, and then when we look at the literature, this plectin protein is been known, but it is a structural protein found inside the cell. Its function is to connect the other proteins. So then we thought we have a roadblock. We messed up something because we were targeting cell surface, and plectin should be on the surface. And of course, there is a paper showing this plectin protein may be coming to the surface when it is in some cancers. But we found this is as a new biomarker for cancer stem cells. So that is, and then we showed that it is found on the cell surface. And then we showed that when you knock down, it loses the binding of our compound and the antibody. And then we showed that when we knock down this protein, it actually blocks this stem cell growth. This is called colony formation. It survives and it affects the survival of these stem cells. And also it blocks the migration, the metastasis. So this is a wound healing assay we call. We make a wound and let the cells to more migrate. And when you knock down this, it actually doesn't do the migration. So it affects the metastasis. And then when you look at the patient data, it actually shows plectin expression is correlated with bad survival. So the patients with high plectin has less survival. So, and then we check the activity when upon dimerization of our compound, it actually kill the cancer stem cells. And you can see it actually blocks this colony formation and destroy these colonies. And also it actually blocks the cell migration. And also in animals, it actually blocks the tumor growth. When we treat with our compound, this is the tumor growth curves, tumor size. This is without the compound, and this is with compound. It actually blocks the tumor growth and without the toxicity, because we found it very specific for that. So as a summary of this project, we found this new compound targeting this cell surface plectin. And it will, if we follow the conventional process, the conventionally biologists will not think this protein come to the surface. And with our technology, it was able to find these biomarkers even moving around. So, and then actually we publish in the scientific reports in the Nature, and it become actually top 100 downloaded papers in the Nature research, and we now showed the activity as well. Okay, let me jump into the third project, and that I going to talk to you in this unbiased applications of our technology. Now here, I am doing the very simple thing, targeting cancer cells over the normal cells. Again, think very simple. So the idea is we are going to very blindly target cancer cells and then identify a compound that doesn't bind to the normal cell. Okay, so without knowing the biomarker again. So we had this very nice cell pair from our collaborator at UT Southwestern Medical Center. Director of the Cancer Center, Dr. John Mina, and he has actually the second largest cancer cell collection in US, and it has been open to me, and I'm very glad. What I did was again stain these cancer cells in red, normal cells from that same patient, same lung. In green, it has to be same patient because the genetic variation shouldn't be there. Only differences should be cancer situation. So, and then mix 1-to-1, exposed to these beads carrying these peptoids, and then we only look for red cell bound bead. So what does it mean? The compound, the peptoid on that bead binds to some specific biomarker found in cancer, which is not found in normal cells. And the other good thing is it can be a protein, lipid, carbohydrate. There are not very good tools to target lipids or carbohydrates. So we did the assay. Again, we found a compound, and it's very specifically pulled out cancer cells, not the normal cells, and we quantify the binding. And again, we don't know the target, right? Like we are doing the reverse way of this drug lead discovery. Now we need to find the target. So I showed you in the previous example, what we do is we let our compound to bind, trigger cross-linking, and pull down. We have been trying this for about a year. So we didn't find anything. So that's again a roadblock, but I didn't give up. Then that's a good news as well, because our compound may be binding to a lipid or carbohydrate, which is very difficult to do in drug discovery because conventional tools are not there. So I thought about it, and then when you look at, there are 4 types of lipids: the phosphatidylcholine and sphingomyelin in the outer layer, this phosphatidylserine, and then PE, PS and PE in the inner layer of the cell membrane. And then Professor Philthorpe has reported that in cancerous situation, this PS come to the cell surface. So he has been very senior professor in this area, and he know my screening technology. He was asking me to target PS because this is a very interesting biomarker. But I couldn't do that because I was very busy. And then when I found this compound, looks like binding to a lipid, I sent my compound to check, and he immediately checked the cell pair that we checked, and he found the cancer cells, of course, highly PS positive, normal cells don't. And then this is the data he sent me. Our compound very strongly binding to this PS, coming to the outer surface. So basically, this yellow are the lipids found in the inner side of the cell membrane. But in cancer, normal cells, in cancerous situation, it comes to the cell surface. So this is a difference between cell surface. That's what our screening technologies very sensitively pick. And then what happened was our compound bound to that PS, found on the cancer cell surface. And then the biggest, coolest thing is, this is the normal PS for, I mean, the lipid found in the normal cell, phosphatidylcholine, and our compound doesn't bind that. Because in the two-color assay, if our compounds, whatever the compounds bound to these blue ones, will bound to this, so we see the green. So we didn't pick them, right? So that's the idea. Anyway, so this has been a very, I'm going to go through rapidly, and even though the protein biomarkers are very heterogeneous, this lipid PS has found in almost every cancer. So this is a can be a global biomarker. So, and when we have these cell surface targets, we always try to do the dimerization because it can improve the activity of the compound. Because if you try to pick something from a single finger, it doesn't work. If you use 2, 3, 4, 5 fingers, it can grab well. So that's the idea. And we do these multimerizations, and my lab develop a fully on-beat synthesis protocols, and we publish them. And we use that, and when we dimerize this compound, what we found was it actually start killing cells. That's great, because our compound not only bind, it actually kills cancer cells, and then it doesn't affect the normal cells very cleanly. And then we in mice, we found our compound shown in red here, very strongly accumulate in the tumor compared to the controls. And very interestingly, again, in the tumor burden assays in the preclinical animal levels, this is the saline treated, where no compound, the tumor grows rapidly. And this is a normal chemo, docetaxel chemo treatment, and this is our compound. And it works same way as the chemotherapy works. This is the combination therapy. Now, it has been reported that when you treat with chemo, this PS flipping increases. That means we have more biomarkers. So that's what happened here. And then again, I'm a chemist, right? So we do a lot of, actually in other projects also we do that. I'm actually showing some chemistry. So we find the most important portions of the molecule that binds to the receptor, and then we use those remaining residues to further improve this molecule, and the dimerizations, the trimerizations, multimerizations, and there are a lot of studies going on there. So as a summary, again, we want to target cancer cells, and we simply compare cancer cells versus normal cells, and we found using our two-color assay, and it only binds compound that only binds to this yellow shown target, which is normally found in the inner layer of the cell membrane. So when this compound is given as a drug, it will target only cancer cells. It doesn't bind to the normal cell. So that's the idea. All right, so let me talk about in the last few minutes the final project that I'm going to talk to you. This is also another very interesting project we started recently. Again, this unbiased application to tumor transform cells from normal to tumor. So this shows the normal tumor growth time with the number of cells. and it's a sigmoidal curve. Initially, it goes slowly, and then this angiogenic switch happens. Remember, I mentioned the blood vessel formation. When the blood vessel formation happens to the tumor, it get all the nourishment, so it grows very rapidly, and then it goes like this. But very unfortunately, the current diagnostic levels are after this angiogenic switch. It is called angiogenic switch. So, but by that time, it's very mostly difficult to control. So, how we can identify a tumor as early as possible, early diagnosis. So to do that, we need to find biomarkers that are expressed in tumor cells so we can target, send these diagnostic agents so then we can get the identification, diagnosis. But so the tumors don't grow overnight. So normal cells become a tumor, maybe even many years, decades. So these initiation, promotion, progression happens. These are called earliest time frame of the tumor growth. And then after it become a tumor, it goes through this angiogenic switch, and then it become a very rapid growth, and also the metastasis happens, the spread. So if you want to identify a tumor very, very early stage, now we all know there's no direct cure for cancer, right? But if we can identify cancer early, we can almost do cure patients because we know what to do at the very early stage. To do that, we need early biomarkers. So how we can find? In conventional ways, okay, you look at the biomarker, now known biomarkers, and then find whether they are found in the initial stage. But I had a different approach. So what I did was, again, very simple thing. Took the normal cells and introduced there are three key mutations that drive a normal cell into tumor, P53, KRAS, and c-Myc. So we introduce one mutation at a time to these cells. And what happened? When these mutations working inside the cell, transforming into a tumor, there can be different new things appearing on the cell surface. It's like, okay, you have a normal person and you poke, you make that person angry, and then the anger come out, right? And then we detect, okay, this is an angry man and this is a tumor. That's what we are trying to do. Again, very simple thinking. What we can do, we have two-color assay that sensitize these cell surfaces very efficiently. So we can compare these normal cells versus these cells, right? These different pairs. So in the first application, we compare these fully normal cells to these tumor transformed cells. We push these cells to become tumor, and these, whatever appearing on the surface, are actually earliest biomarkers. We simply let the thing to happen and then pick what is coming out. That's our purpose, and that's natural. That's what. So mostly these biomarkers would have been the real biomarkers in the early stages. So that's what we are going to pick. So we did the assay with the normal cells in green, the tumor transformed cells in red, mix 1-to-1, expose to the library, and look for a bead only bound with the red cells. That means the tumor cells. That means the compound bound to something on the cancer cells that is newly appeared compared to the normal. And then we pull down, and again, we did the pull down like we did in the early, and found that this vimentin protein as the target. It has been known, but there were not much studies done in the early phases. So after finding this, there are two ways we can go, diagnostic and therapeutic, and this is the diagnostic area. And we showed that our compound can very strongly binding to the tumors compared to normal, and that is compared to the conventional antibody. This is actually histology screening. And then we are in a very, very early stages of trying to develop simple diagnostic tests using this. I'm not going to go to the details. And then again, you all chemists, I'm sorry I didn't have much chemistry slides here, but we found the most important residues of this compound, and then we try to further improve in multiple medicinal chemistry approaches, like fine-tuning the important residues to bind better, and then convert these non-binding residues into binding modes, and identifying new binding pockets outside the other binding areas, and also dimerizing to get the avidity effect. And then we test this in a very interesting cell systems. So there are high vimentin expressing cancer cells, moderately expressing cancer cells, and low vimentin expressing cancer cells. And what we found was our compound is very effective in high vimentin expression. So that indicates it targets the vimentin, and it has less effect on the less vimentin expressing cells. And again, it actually destroys this colony formation, this tumor cell survival, and also it blocks the migration as well. So as a summary, in our assay, what we did was we compare normal cells and then force them to become cancer cells, and then identify whatever appeared here. So this is, again, I told you at the very beginning, the conventional way is you biologists first try to find this and go back, but what we did was the alternate, okay? And by doing that, again, we did a very big thing because we identify new biomarkers and also drug leads together. We target big, but it's very simple. Simple thinking, two-color comparison, and then we stayed focused. So that is the take-home message I want to give throughout my story as a scientist. So here actually I'm summarizing everything. In this screen, we found so far 7 protein targeted compounds and 1 lipid targeted compounds. All these 8 are patented or patent pending. And then actually we have a lot of imaging applications and also theranostic agents, which has both therapeutic and imaging capacity. That means we can do monitor the therapy. That's the idea. Anyway, I didn't have a chance to talk any of this. So this is the drug discovery process: initial drug lead identification through screens, hit validation, optimization, and then we get into the cellular and animal levels, and then clinical. So these are about 9 projects that are going on in my lab, and these are the only 4 I have shown here. And you can see, I mean, the green means we already completed, and these are this is the current status of these projects. They are mostly in the preclinical levels. And finally, I want to thank all these people, and without their support, I wouldn't have done any of this. So all my lab members, the very, very important collaborators, and also funding. Without any funding, I wouldn't have done anything. So the NIH, CPRIT, and there are many other organizations and the companies funded me. So I think that's it, and I would stop here, and thank you very much for your patience, and probably I'll go back to the summary, and I'm happy to answer any questions.
Okay, thank you very much, Professor Gomika, for your wonderful talk. I'm pretty sure that everyone is really inspired by your talk, and some of the topics are really relevant to the current situation. I mean, the COVID situation is again getting worse in our country as well. I mean, I know it's the same thing in the USA as well. So I'm very sure that the audience will have so many questions, but with all these time limitations, we will try to have many questions as possible. So I would like to give the chance to the audience to come, have some questions for Professor Gomika. Thank you very much once again for your wonderful talk, Professor Gomika.
You're welcome. Thank you so much.
Thanks. Questions, the audience?
Professor Udugamasooriya, I'm Dimanti. I'd like to know, first of all, thank you very much for that interesting talk, and you presented it in a very simple way, easy to understand way. Thank you very much for that. So the peptide synthesis, how do you determine the length of the peptide synthesis, and like how do you determine which amines to bind, and is it from previous hits, or how do you determine that?
Yeah, that's a good question. Okay, so the length, typically you can see my slides, right?
Yes.
Okay, so the length we decide based on where we target. So we need at least about 4 or 5 residues for a compound to bind, because these binds like this, right? Like a chain. So these side chains getting into the binding pocket. So for cell surface receptors, they are like big proteins. So we need about actually 6 to 8 or 10 residues. That's how we decide that. We can go beyond that. That's fine. And then to answer your second question, the amines we decided, yes, most of them we decide based on our previous work. Initially, actually, I told you, my postdoc advisor, Professor Koderek, shown how these are how biologically important, and there was some set of amines that were validated. They are very important in binding in proteins. So, for example, like this amine, I mean the lysine-like, natural amino lysine side chain-like, and then leucine-like, tryptophan-like. We have those mimicking exactly amino acid. But the cool thing is we can bring all these different organic moieties. They can actually, so the coolest thing about this peptide is it can bind new hotspots on proteins that the natural proteins or amino acids cannot do. These are new synthetic moieties. So yes, so and then when we get introduce new amines, we actually validate them as well.
All right, thank you. And I have another question. So the two-color binding you showed, can there be competitive binding as well?
Of course, great question. So what we do here is, okay, the initial idea is to identify a compound targeting our most interested protein to be blocked, right? And then it shouldn't bind to any other places. But at the same time, we can actually, of course, do that. Actually, we had some interesting thoughts. Most of the time, we want to antagonize a receptor, right? So block the receptor. Sometimes it is important to agonize, trigger the activity on some diseases. So how do we do that? Now, when the protein, the compound or the normal hormone binds to the receptor, it undergoes different conformations. So if sometimes we can actually lock them in different conformations and then do the screen. And also, yes, do the competition. And there are different applications you can do. For example, if I show you a direct competition, when we improve the binding, let's say this was the initially identified compound, and we want to find more binding hotspots. So we can have this initial compound already bound to this area, it blocks, and we develop the library with more residues. So we competitively block this part, and then we find new binding areas of the protein. Did you get that?
Yes.
Okay.
All right. Thank you very much.
You're welcome.
More questions, the audience? Anyone from the audience?
There's a question in the chat box also.
Okay, madam.
It's from a first-year student.
I cannot see
It
anything.
says, if I just read the question, it says, is drug industry mainly based on chemistry or biomedical science?
Okay. It's a hard question. I mean, again, normally what happens is In the academic level, we find the initial drug leads and then validate to certain level, and then the industry pick that, and then do the, because for the clinical trials type work, it's very hard for academic level to fund them. So one way is those academically developed compounds getting into the industry, and they validate them in these expensive clinical trials. The other is actually they also have R&D arms always. I mean, they do the initial screens, and especially in this high throughput screening, the industry does very initial hit identification to the whole process by themselves as well. The both are happening, and they have very highly focused synthetic, organic, and chemistry type arms, and on the other end, the clinical level biomedical validations. So it's hard to say only one thing happened, but both are happening. But mostly they focus on the final stages of the drug discovery. I hope I answered the question.
I think there is another question in the chat window. So when was the first place you thought that you should concern about biomedical science?
Okay. I don't know. I have to think. Like I said, from the very, very early stages, as a small kid also, I have been doing research, and then, yeah.
Yeah.
As an undergrad, and actually doing my project with, like I said, my great master, Prof. We were doing some isolation of compounds, some algae, and then look at the biological activity, and that actually very trigger. I mean, it triggers my interest into the real application level, because he had collaborations with the US and Canada, and then we again there's another big thing here. We were not just only doing the isolation characterization of the compounds, we were really focusing on the final application. So he sent those immediately to Canada and US and validated. I'm talking about this like more than 2 decades back, and so it was huge. So I think all those helped me to trigger these applications, and again, I said, I mean, I am actually, so please don't misunderstand. I'm doing a lot of chemistry, a lot of synthesis, and I didn't show all those in detail, but my always thinking to apply it and what we can do to the society, especially in my case, biomedical applications. And then, of course, you have a lot of different, right? I mean, the analytical, surface chemistry, nano, even computational, there are a lot of different applications. Whatever your field, think about what is the impact you can bring in. And then actually it helps to do many good things also in the chemistry level. Remember, I showed you many different optimizations. So we come back and refine our molecules that can bind well. So for example, if it binds here, we try to bind to another place as well. That's purely chemistry. It's chemistry knowledge, but it can do very big application in biology, because that additional binding pocket may be not found in other places. So as chemists, we need to really, this inspire, I like this seminar series, and you need to find the scientists inside you, and we all have. It's only about finding them.
Okay, thank you very much, Professor Gomika. I mean, it was a detailed explanation about what has been asked. And to add to the same question, what is the secret behind your motivation towards doing all this stuff? We all together, like all the senior research people here here at Pera, as well as the young researchers, we all inspired by your talk. And there's no doubt that you're a really hard worker, a great worker. So what's the secret behind that? How did you get that motivation from the beginning? Just a short response would be helpful for our young researchers. Thank you so much for your time.
Exactly, the great question. So again, like I said, we always need to go beyond, come out and challenge the world. That was my motivation. I always challenge the world. And actually recently I was given the same kind of a talk in SLAS, and there were some questions about our system, education, and I said there are a lot of things need to be improved, and we need to change that, because there are a lot of talent being misplaced in Sri Lanka. We all have a lot of talents. So my answer was yes. I mean, I challenged the system, and I didn't follow the status quo. So that's my success. And I always wanted to go out and do something new. Again, don't think that's a hugely extraordinary thing or extraordinary capability. No, we all have it. And I'm sure, I can assure you, you all have that. Please find that. Come out and then challenge the normal things happening. And all the new development, research, new findings, they all came because you didn't stuck to the whatever the box your comfortable zone. You need to come out of it and then look for new things. Then you see how big the universe, right? And how much new things you can find. But again and again, I'm telling you, don't make things complex. Target big, but think simple. And I keep telling that if you throughout look at all the major developments that I did, and they are very, the screening assay, and there are many other places using, and then we just completed the Peptoid International Conference. I always invited to give a talk. So there are a lot of interesting developments and changes we have made. And but again, it's all because I didn't stick to the comfort zone. So you need to come out of that comfort zone and just think to doing that, and then you will find a lot to do. And I don't know whether I answer your question, but I don't know how it happened, but it happened.
I think you answered. Yeah, thank you so much. Okay, once again, thank you so much, Professor Gomika, for your very inspirational talk to us, for both young researchers as well as senior researchers. So I think we spend more than 1 hour. It's about now 1 and 30 minutes. I'm pretty sure that Professor Gomika is really busy and he's having other stuff to do in the morning as well. So, okay, so I would like to move to Mr. Fiham.
And I wish you all the best. And also, if there's anything that I can be helpful, please let me know. And then I never forget the free education I get from Sri Lanka, and then I do my best to pay back. And so I'm ready to help in any way. So if there are anything that I can be helpful, please let me know.
Thank you so much. We hope you will spend a few more minutes with us.
Yeah, thank you so much. Yeah.
Good evening, everyone. I have been honored with the privilege of presenting the vote of thanks on behalf of the Department of Chemistry of University of Peradeniya and members of the Inspire team. First, I would like to express my sincere gratitude to Professor Gomika Udugamasooriya for accepting the invitation and spending his valuable time in sharing his story on his contribution to developing technologies to advance
I think he's having.
Discovery. Thank you very much, Professor Udugamasooriya, for your inspirational webinar. In my sincere gratitude, Professor Manadevi Ganahenage, head of the Department of Chemistry, for hosting. And I would like to thank the attendees for being with us this evening. I'm sure you would have found this webinar very informative and inspirational in shaping your academic future. Thank you. Have a pleasant evening.
Thank you so much, everyone, especially to Professor Gomika.
Thank you. And good luck to everyone.
Thank you so much.